Today we'll be talking about glycogenesis and glycogenolysis. So the main thing we need to first know about is glucose. How do we get glucose? We get glucose through a process called gluconeogenesis, in which case we're going to convert pyruvate into glucose. And it's mostly the same steps and enzymes as glycolysis, but there are a few differences. But basically, we have this glucose. But what's going to happen if we have too much glucose? So if there's an excess of glucose, it's all going to go into the liver, and it's going to get stored there. So this glucose is going to get stored in the liver in the form of glycogen. Now, this is where glycogenolysis will occur. We're going to take a bunch of sugar molecules and we're going to put them together to get glycogen. So when we make linear chains of glucose like this, these are called alpha 1,4 linkages. And I like to think of it as alpha 1,4 goes forward. So it's going forward. It's just a linear process. So we're adding to the chain of glucose. If we're going to make branches, and then a chain again, so this, again, we have alpha 1,4 linkages, but where we got this branch over here, that's an alpha 1,6 linkage. So a way to remember this is 1,6 throws a branch into the mix. So branch into mix. So 1,6 throws a branch into the mix. And the reason that we're adding these different branches to glycolysis is that to glucose and glycogenesis is that we have these ends of the branches which are available sugars that we can break off in case we need it. So we have liver, and then we have the bloodstream, and we have our brain. Our brain needs glucose to function properly. So if there's not enough glucose in the body, so if we have low glucose, then that's going to cause glucose to start breaking off of the glycogen stored form of glucose. So on these branches, they're going to break apart, and when they break apart, it's not going to be exactly the form of glucose, so you have to convert it into glucose 6-phosphate first, and then you can convert it into glucose. And that glucose is going to enter the bloodstream, and it's going to enter any of the cells we need, but mostly the brain, because the brain needs glucose. Now, if we've really totally run out of our glucose stores, and we have really, really little glucose, that's when our pancreas is going to release something called glucagon. Pancreas is going to release glucagon into the bloodstream, and glucagon is giving the sign that glucose is gone. So glucagon equals glucose gone. That's kind of a good way to remember it. And if the glucose is gone, and we already depleted all of our glycogen stores over here, that means we need to go through gluconeogenesis in order to make more. So we're going to want to do gluconeogenesis, but in order to do gluconeogenesis, we need a lot of ATP. And that's where our adipocytes, which are cells in our body that store fat, our adipocytes are going to release those triacylglycerides. These are the storage forms of fat. It's 3 fatty acids bound to a glycerol backbone, a glyceride backbone, and they're going to enter the bloodstream, and they're going to enter the liver, and they're going to enter the mitochondria of the liver hepatocyte. And now, in order to get ATP, we can start breaking down these fats via beta oxidation. So beta oxidation is basically we're going to take our fats and we're going to break it down into acetyl-CoA. And while we do that, we're also going to release FADH2, which gives us 1.5 ATPs. And we're also going to release NADH, which gives us 2.5 ATPs. And then we also have our acetyl-CoA, which gives us 10 ATPs. Now, if we add all this up, we have 10 plus 1.5 plus 2.5, and that's 14 ATPs that we just released. That's a lot of ATP. And in order to make even more ATP, because we have this acetyl-CoA, we can get this acetyl-CoA into the citric acid cycle. So then we can get our reduced cofactors into the electron transport chain, and then we can make even more ATP. But the thing is, because we're using up energy to make gluconeogenesis, and in order to make gluconeogenesis happen, and in order to get acetyl-CoA into the citric acid cycle, we need to convert it into oxaloacetate first, right? You should remember that. We need to get oxaloacetate out of acetyl-CoA in order to get the citric acid cycle moving. However, since, as I mentioned before, we already have a bunch of gluconeogenesis happening in the liver, that gluconeogenesis is also using oxaloacetate. So we can't really go on to gluconeogenesis to make more sugars, because, we ran out of sugars. That's the whole reason our pancreas released glucagon in the first place. So now is where we can use this acetyl-CoA to form ketone bodies. And we can form keto acids and ketones, and those are going to enter the bloodstream. Those keto acids and ketones, they're going to enter the bloodstream, and they can enter any cell that need them, and these cells can use those ketones as an energy source as well when there is not enough glucose. So now we have ketone bodies giving us the energy we need in place of glucose, and we also have a bunch of glucose in our body because after we released glucagon, we counteracted that. Now we have enough glucose again. But if we have too much glucose, if we have way too much glucose, that's when the pancreas is going to release another important hormone, and that is insulin. So the insulin is basically going to keep everything in check. Insulin and glucagon are going to work together to make sure everything's in check. So that's why for patients with diabetes, either diabetes 1, in which there's no insulin being produced, or diabetes 2, in which there is insulin being produced, but the receptors are inactive towards insulin, that's going to cause a lot of problems with mediating our blood sugar and keeping it in the proper place. So that's why glycogenesis and glycogenolysis is really important, because it helps us control how much sugar we're storing and how much sugar we're releasing into our bloodstream. And based on the hormones that are released by the very important pancreas, it determines how the course of glycogenolysis or glycogenesis is going to occur.